Hi, my name is Dr. Edwin Kim, and I'm an Associate Professor of Medicine and Pediatrics at the University of North Carolina School of Medicine. I'm also the Director of the UNC Food Allergy Initiative Research Group. And today I'll be giving an update on the FAIR-sponsored study, Peanut Sublingual Immunotherapy, Induction of Clinical Tolerance of Newly Diagnosed Peanut Allergic 12 to 48-month-old Children. First, I'll start with a few of my disclosures. And I have consulted and received honoraria from both Immune Therapeutics as well as DBV Technologies, and I act on the Clinical Medical Advisory Board for DBV Technologies. I have several other consultancies and have received federal as well as private grants for our research. Sublingual immunotherapy represents a convenient and a painless form of immunotherapy compared to allergy shots. It's administered as drops that are held under the tongue, in our case for 2 minutes, and then swallowed. Of note, the doses used in sublingual immunotherapy are 100 to 1,000 times lower than the quantities of allergen that are typically used for OIT today. Another important point is that there is no associated food taste or smell with food sublingual immunotherapy. It is important to note, though, that at this point, sublingual immunotherapy is approved for aero allergens with specific formulations, but it remains investigational for foods. Now, our group has been studying sublingual immunotherapy for almost the past decade, and last year in September, we were able to present or publish our long-term results of sublingual immunotherapy in 1 to 11-year-old children. And here, what we used was a 2 milligram peanut sublingual maintenance dose and treated children for 3 to 5 years. At the end of that treatment period, we did a food challenge to 5,000 milligrams of peanuts, so the approximate equivalent of about 16 to 20 peanuts. The median tolerated dose within our group was 1,750 milligrams. And of note, almost 1/3 of these patients completely passed the food challenge with no symptoms at all. Of those 1/3, we further removed the treatment for 1 month and had them return for a food challenge to look for a lasting effect. And what we found was 27%. So 10 of those 12 patients were still able to pass their food challenge 1 month after stopping therapy. The summary we had from our results was that SLIT represented a very easy to do treatment, and there was very good compliance to back this up. The safety was also reassuring, with the primary side effect reported being oral itch that self-resolved typically within 10 to 15 minutes. Symptoms requiring more advanced therapies such as epinephrine were very rare. We saw clinically significant desensitization for the entire cohort, but when we looked at subject to subject, there was a very wide range of results, from some patients passing with no symptoms to others looking no different than placebo. Now, one question we wanted to ask as a group was, how do we actually make SLIT better? When we looked at studies of OIT and epicutaneous immunotherapy, there was a suggestion that perhaps intervening early when children are younger could have an improved effect. Specifically looking at 2 studies published by Brian Vickery, one of our FAIR investigators. He first published on a combined OIT study between Duke and Arkansas and demonstrated that patients who had a lower IgE at baseline tended to have a better result, including a higher chance of a sustained unresponsiveness 1 month off of therapy. He used this information to create the DEVIL study, which looked at 9 to 36-month-old children, with the thought that these younger children would be more likely to have lower IgEs at baseline. In his study, he found that nearly 80% of this younger cohort was able to sustain their desensitization or represent the sustained unresponsiveness 1 month off of therapy. 2 industry studies also demonstrated an age effect. The phase 2 VIPE study studying the DBV Technologies peanut epicutaneous patch seemed to demonstrate that the younger cohort in that group, ages 4 to 11, had a stronger effect than the children that were older than 11 years old. The phase 3 PALISADE study run by Immune demonstrated a strong effect in the 4 to 18-year-old range, and that has resulted in FDA approval of their product. But whether because of numbers or because of a lower effect, an effect in the older group was not seen. So could there be an age effect in PnSLIT? To date, there have been 3 primary studies looking at PnSLIT in allergic patients. The first study being the COFAR-4 study, followed by a Johns Hopkins study, and then the long-term study that I just described that we ran at UNC. And here in this table, you see the different tolerated doses, with COFAR-4 representing the lowest tolerated dose, 371 milligrams after treatment, followed by Hopkins at 496, and then our study at 1750. When we look at the ages of the patients that are in these studies, what you'll see is that the COFAR-4 study by far had the oldest group with a median age of 15 years of their patients, followed by Hopkins at 11, and then our study at 6.5. And this suggested that the degree of desensitization could potentially be affected by the age at baseline in entering these treatments. This led us to the development of our FAIR-sponsored PnSLIT study. And the primary questions we wanted to know were, can younger age improve the outcomes with PnSLIT? Can earlier intervention with PnSLIT produce a longer-lasting treatment effect? Ultimately, we started the peanut sublingual immunotherapy induction of clinical tolerance of newly diagnosed peanut allergic 12 to 48-month-old children study. And the clinicaltrials.gov identification number is here on the slide for you. This is a 2-center collaboration between 2 FAIR center studies, our center here at UNC, as well as Dr. Drew Bird's center at UT Southwestern. Here you see the protocol in figure form. And in this study, what we included were peanut allergic children ages 12 to 48 months who had a strong clinical history of a food or a peanut reaction, as well as a positive peanut skin prick test and a positive peanut specific IgE. All patients had to undergo a baseline entry food challenge and have reactions at less than 1,000 milligrams to qualify for the study as well. At baseline, patients were then, once qualified, randomized 1-to-1 to receive either PnSLIT with a 4 milligram maintenance dose or placebo for a 3-year period. In this diagram, you see that there are every 6-month visits at the clinic and annual lab results as well. After 3 years or 36 months of total treatment, patients undergo a desensitization, double-blind, placebo-controlled food challenge to 4,443 milligrams. After this food challenge, the patients are then instructed to withhold the PnSLIT or placebo treatments for 3 months and then return at 39 months of the study to repeat that food challenge to demonstrate or not demonstrate a sustained effect of peanut sublingual immunotherapy. At this point, we can show you some of the baseline characteristics of the patients. So what you see over here in these 2 tables, first of all, this is a very young and atopic cohort with a median age of children of 2 years, a male predominance as well as a Caucasian predominance, which has been very common in our area. And many, if not the majority of patients, having multiple food allergies and eczema, as well as a subset having asthma and allergic rhinitis. When you look at markers of sensitization, the skin prick tests, as well as the peanut-specific IgE, what you see is a wide range, despite being as young as 2 years of age in this study. And at baseline, patients reacted at about 286 milligrams, or tolerated 286 milligrams during food challenge in order to enter into the study. Enrollment began in January of 2015 with a target goal of 50 participants, 40 at the central UNC site and 10 at the Southwestern site. We were fully enrolled and moving along with the study, and then unfortunately, like everyone else has experienced in the world and in our country, coronavirus had hit. Protocol modifications were made as of March 2020 to adjust for the COVID-19 pandemic. Patients were allowed to have remote maintenance visits, and any lab tests or skin tests that were due were deferred till the next live visit was allowable. We received approval to continue SLIT maintenance dosing for any subjects that were due for a food challenge, and 6 subjects extended treatment beyond the planned 36-month treatment period while awaiting their food challenges. Research reopened fully at UNC in June of 2020, and we've continue to move forward with our study. And at this point, 2 subjects remain, both of whom are in the avoidance stages of the study. So there are no longer any patients that are receiving PnSLIT at this point. We anticipate study completion actually next month in October of 2020. Once we're able to get the last couple patients through and pass their food challenges, we will analyze the data and lock the database, and our next steps will be hopefully an abstract submission, a late-breaking abstract submission to the 2021 Quad AI meeting, as well as a manuscript in early 2021 as well. We're excited about the potential effects of PnSLIT on this younger cohort and look forward to these results. And finally, I want to conclude by just being able to say thank you and showcase the broad group at our UNC Food Allergy Initiative that's been responsible for not only this study, but many of the other studies that we've been able to conduct, including our physician investigators, of course, Dr. Wesley Burks, who has been a world leader in food allergy immunotherapy and has helped mentor and train me to get to this point of directing the group. Allison Burbank, Scott Cummins, and Onyi Wala, who helped me to conduct the food challenges and the other visits as investigators. Our allergy fellows, who are intricately involved in the oversight of our trials as well. Our study coordinators, led by Lauren Hurley, as well as our 4 other coordinators who really do the yeoman's job when it comes to these food studies. And in particular, I do want to call out Lauren Hurley, who was really the primary driver for recruiting for this very, very difficult 3-year placebo study. We're fortunate to have a GMP manufacturing group that helps to produce the PnSLIT therapy, as well as a basic science lab that runs the mechanistic biomarkers, as well as does novel science in food allergy as well. And with that, I thank you for your attention.